is your educator, Dr. Shivangi Shukla. I am your educator for biochemistry here. I'm currently conducting a very rapid revision of the entire course of biochemistry, and this is stipulated to be over within this year. So before 31st of December, we shall have revised the whole of biochemistry, which we have already covered within 1 month in the crash course. Okay, so twice in a very rapid manner, we shall be covering the whole syllabus of biochemistry. Okay. Alright, so let me just check if I'm visible and audible to all of you. Let me just check if everything's clear and good enough to start. Okay. Great. So now let us begin. Alright. Today we shall be discussing about extracellular matrix. Now without further ado, let us quickly discuss about the types of subscriptions for NEET PG that are available on Unacademy, on the Unacademy app. They are iconic as well as plus. The iconic subscription is an additional, it has additional resources to plus. Now what does the plus subscription provide you with? It provides you with access to India's top medical educators through their live as well as recorded videos, their live quizzes, regular updated integrated sessions, integrated quizzes with different teachers, different educators. You also get to access the printed notes in the PDF format and regularly updated question bank. Also, you can access this while on the go on any device of your choice. So this is the USP of this, alright. And the iconic subscription comes with access to extra resources from Prep Ladder as well. Okay, so it is rightly termed best from two of the best. Okay. Do not forget to put in the educator code DRSHUKLA, Dr. Shukla. This code is going to fetch you an additional 10% discount. So please guys, do not forget to put in this code. Okay, now tonight we shall be discussing about extracellular matrix in 2 separate sessions. Okay, very quick sessions and extremely important topic. We have discussed this twice in our crash course, and we have discussed the structure of collagen in great detail. But tonight we shall be revising all of this. Okay, so that it gets ingrained in our memory. Okay, now the next time when we'll be discussing it, I hope you guys teach me something about it. Okay, in the comment section. Right now, okay, just a brief description. I take classes on Unacademy special classes on Unacademy app. You guys have the app, well and good. Do check me out in the section. Also, I take classes on this channel called Unacademy Future Doctors. This is a new channel launched by Unacademy, especially dedicated to MBBS Prof 1 and Prof 2. I am your educator for biochemistry, and do follow my earlier classes if you haven't seen them. Do go and check them out. Also, I have been taking regular tests, regular answer writing sessions, as well as MCQs. Okay. Tomorrow there's going to be an MCQ session on Unacademy app. Tomorrow 9 AM in the morning. Alright. MCQ session, that is the 5th in the session. That is a series that is called MCQ Bonus Biochemistry Sessions. MBBS. That's an acronym. Okay. That's the 5th in the series. 4 already we have done. So that's a weekly contest for you. Okay. So do enroll yourself in that class. It's absolutely free. Go back and enroll yourself in the class and do attend that tomorrow morning 9 AM. Okay. Now let us start with extracellular matrix. Now what is the extracellular matrix? In common parlance, it is the connective tissue. See guys, this is a revision class. Already 4 times we have discussed extracellular matrix in the last 1 month. So I'm going to be real fast here. I'm just going to discuss the important steps, the important terms and the mnemonics that we have discussed so that it gets ingrained and consolidated in your memory. The extracellular matrix is the connective tissue. What does it have? It has 3 classes of biomolecules: the structural proteins, the specialized proteins and the proteoglycans. The structural proteins are of 3 types: collagen, elastin and fibrillin 1. Following, what are the 3 types of structural proteins? Collagen, elastin and fibrillin 1. We are going to discuss each of these, the structure of each of these, as well as the diseases associated with deranged structure. Okay, all right. Second is the specialized proteins. They are of two types mainly, fibronectin and laminin. Okay, fibronectin and laminin. We are going to discuss the structure as well as the location where they are present. All right, and the applied clinical applications like any disease states or any defect in the structure and all. Okay, all right. And the third one, the most important, are the proteoglycans. The proteoglycans are what? Proteo plus glycans. So they are complex polysaccharides containing protein. Okay, glycans are what? Polysaccharides, carbohydrates containing proteins. Okay, now so we'll discuss about them, and the applied concept that we are going to discuss about these are mucopolysaccharidosis. So in the first session that is going on right now, what are we going to discuss? We're going to discuss about two major biomolecules, extracellular matrix, that are collagen and elastin, and we're going to compare them. Okay, now this, like the revision class, the earlier revision, the rapid revision classes that we are conducting, that I am conducting, the rapid revision classes, there is a pattern where what do? We try and behave, we pretend as if this is a simulated, this is a classroom environment or examination hall setup, right? This is what we pretend. So now what we are pretending is that we have got a short note or we have got a long question on collagen. Okay, and then we are going to write the answers. How are we going to write the answers is my main focus. Okay, because when you come straight from your PM, after you crack your PMT, writing answers in school. Now there's a shift. There's this drastic shift when you have entered college, and there's something else expected of you to write, to answer in the professional university examination. Okay. Now, that is the most important thing, is that your answers should be illustrative. Illustrative, not just with diagrams. They should be abundant in points, in tables. Okay. It should be it should not be a paragraph kind of wrote, like no one likes to read long paragraphs or long sentences. Okay? So similarly, the examiner also doesn't want to read long sentences. What the examiner he wants to know the keywords. So my focus is always on the keywords, and my slides always my slides contain what? My slides do not contain any printed material. I always make sure that I draw every diagram by myself. I write all the answers by myself, so that you guys know how to do it. You guys can get confidence of doing that in your exam as well. Okay, so let us begin with the structure of collagen. Collagen, about a brief introduction you can give, because every answer is well structured. Every answer needs to have a proper structure. So you should think of that structure, that blueprint in your mind from before, all right, and then start writing the answer. You can give about 10-20 seconds to develop the structure of the answer, especially for long questions. Okay, the long questions need to be properly structured. You cannot just go on jumping from one topic to the other. Okay, the answer should be well structured, well formed in your head from before. You can take around 1 minute to structure the answer and write down points, and then make the start writing the fair answer. Okay, so collagen, an introduction is that it is the most abundant protein in the animal world. All right, collagen is the most abundant protein in the animal world, and 25% of the proteins present in mammals are what? Collagen. Okay, 25% of the proteins present in mammals is collagen itself. All right. Now, this is a side of what we are discussing, but it is extremely, extremely important. I cannot stress how important it is for your MCQs, for NEET PG entrances, INI-CET entrances, FMG exams. It is extremely important. Non-collagen collagens. What are these? They contain, these are molecules that contain collagen-like domains. Now, going forward, we are going to read what collagen contains is gly-XY primary structure. Okay, gly-XY, gly-XY, like you can write it as gly-XY N. That means N number of gly-XY. So, this is this generic structure is present in these molecules as well, these non-collagen collagens as well, but they are not typically collagens. They have some other properties, they have some other changes and modifications in the structure. They are not collagens, but their structure resembles that of collagens. Okay, therefore they are called non-collagen collagens. Two very common and very often asked examples are the C1q protein of the complement system, the pulmonary surfactant protein SPA, surfactant protein SPD. SPA and SPD, surfactant protein A and surfactant protein B. Both of these as well as C1q of the complement system they are non-collagen collagens. Okay, very important. Next is the structure of collagen. The structure of collagen is a triple helical structure. What is a triple helical structure? It is a triple helix. That means it contains three separate helices, three strands which are alpha helices, and then this is the suprastructure. Okay, so three helices are left-handed. Three individual alpha helices are left-handed, whereas the suprastructure is right-handed. Why is it right-handed? Because right-handed is more stable. Why is it more stable? Because it has 180-degree or collinear hydrogen bonds. Okay. Now this is what? This is a rod-like structure. What is this rod-like structure of collagen called? Fibril. Now there is another type of structure that is meshwork structure that is present in type 4 collagen. Guys, this is a revision class. You have to be interactive. I am not going to speak out each and everything. I'm not going to feed you spoon. No spoon feed. Now, what is the importance of this opposite-handedness? It resists unwinding of the collagen. Therefore, it provides it strength, the connective tissue strength against unwinding, because it is very strong. So this ringing movement whenever we do of the skin tissues, then the collagen breakdown is prevented through opposite-handedness. Always remember. Okay. Next, this is the structure. So, for every turn, we have 3.3 residues. What 3.3 residues means? Gly-XY, you see. There are almost 3 amino acids, right? 3 amino acids. So, for every turn, you have almost 3.3 amino acids, 3.3 residues. Okay? Alright, and almost 1000 amino acids per chain are present. The diameter is 1.4 nanometers. These are the important terms that you can remember if you're good at it. Okay, so what is the structure? Gly-XY, gly-XY, gly-XY. This is the generic primary structure. Now, this is glycine. Why is every third amino acid a glycine in collagen? Because glycine is the smallest amino acid and it can fit very easily in this tightly packed fibrillar structure. Very tightly packed. So, there's space for only this small glycine there. Okay. Now, X and Y are usually what? Proline and hydroxyproline. And this hydroxyproline and proline, it provides rigidity to the collagen molecule. How is hydroxyproline formed? Through prolyl hydroxylase enzyme. And what are the cofactors? Just day before yesterday, I think we discussed in the chapter of amino acids. The cofactors are vitamin C and alpha ketoglutarate. Therefore, in scurvy, that is a deficiency of vitamin C, what happens is defective collagen. Therefore, there is easy bruisability, there is bleeding, easy bruising, bleeding, swelling of tissues. Okay, got it? Similarly, whenever lysine is present in the Y position, Lysine is also converted to hydroxylysine through the enzyme lysyl hydroxylase. Similar cofactors, vitamin C and vitamin C and alpha ketoglutarate. Vitamin C and ascorbic acid are one and the same. So vitamin C and alpha ketoglutarate, or alpha ketoglutarate and ascorbic acid. Okay, and what does it form? Hydroxylysine. They also provide rigidity to the collagen structure. Okay, now there is another modification that happens in hydroxylysyl group, that is glycosylation, or the addition of a carbohydrate moiety. Glucose or galactose gets added on hydroxylysyl group. Okay, through O-glycosidic linkage. Always remember O-glycosidic linkage. Okay. Now, yeah, so I already told you that hydroxylysyl, hydroxyprolyl, all these provide rigidity. Now, how does hydroxylysyl provide rigidity? Through interchain and intrachain covalent cross links between the collagen structure. This is a revision class. I'm trying to revise. So if at all you have any doubts in the concept part, you can go back and revise the classes that I have already taken. Otherwise, you can put in your doubts in the comment section as well. I'll address them. Okay. Next is an enzyme lysyl oxidase, which has copper as its cofactor. And lysyl oxidase, what does it do? It converts it into aldehyde form, and this aldehyde form also, what does it do? It provides interchain strength and rigidity. Okay, if you guys remember, therefore in Menkes disease, which is the deficiency of copper, what is Menkes disease? Menkes disease is the deficiency of copper. Here what happens is kinky hair, brittle hair. Why? Because of defective collagen. Okay, got it? Now, the collagen molecule has a banded appearance. Why does it have a banded appearance? The collagen molecule, because the different fibrils, they are not arranged just one after the other. No, they're not arranged like this. They are longitudinally displaced by about 1/4 of their length. That is why quarter. Staggering means displaced. So they are staggered by about 1/4 of their length. That is why there is a banded appearance. Very important. Okay. Now I already told you about the meshwork appearance of collagen. This occurs in type 4 and it is present in basement membrane. Okay. Now why does it occur? Because if you guys reckon your mosquito net, it has small holes connected by small strands, like small threads. Okay. So these holes are the places where the GlyXY structure has been interrupted. The GlyXY structure has got lost. So there are spaces. And these bands are what? These bands are the normal fibrils where GlyXY is present. Okay. So this now becomes a meshwork. Okay. Like similar to your mosquito net. Always remember your mosquito net. Okay. Now, how is collagen synthesized? Collagen is synthesized as a pre-pro molecule. What is the pre-pro molecule? What is pre? Pre is is a structure, or pre is a molecule. It is a signal molecule. Okay? It signals the pro-collagen to go towards the endoplasmic reticulum. Therefore, pre is what? The monitor or a leader. Okay, similar leader we have read in DNA replication, that is primer. Primer was also the leader, right? So here the pre-group is a sequence of amino acids which is a leader or a monitor or a guide for the pro-collagen, that is an inactive collagen molecule. And this pro-collagen will then be sent to the directed to the endoplasmic reticulum, where the pre-group will say, okay, bye-bye, my work here is done. I have brought you to your manufacturer. Now the endoplasmic reticulum, what does it do? It is the mentor. Here the pro-collagen, the inactive, the immature collagen will be mentored into a mature structure. Okay. Now here is where in the endoplasmic reticulum this the hydroxylation of proline and hydroxylysine takes place here in the endoplasmic reticulum itself. Okay. Now, why is this pro-collagen immature? Because it contains some extra baggage. Okay, once a person becomes mature, he becomes very relaxed. A mature person becomes relaxed. The mature person has dropped off the extra baggage, whatever it had been carrying on its shoulders during times of immaturity. Now, this immature or inactive procollagen contains some extra extension peptides at both its ends, amino as well as carboxyl ends. Okay, how are these? Now, this is the procollagen. Okay, this is the amino end, the amino terminal, which contains extension peptides. This is the carboxyl terminal that contains extension peptides. Okay, now there are disulfide bonds between cysteine residues on both these extension peptides. Or at the amino terminal, the disulfide bonds are just intra-chain. That is, they are present only on this chain, only on the parent chain, on that particular chain. Whereas on the carboxyl terminal, the disulfide linkages are both intra as well as interchain. You see, this is the triple helix. There are 3 helices. So they will be, excuse me, they will be both intra as well as interchain. Now, this interchain disulfide linkage is what starts the winding of the 3 helices, 3 alpha helices. Okay, therefore it is called self-assembly. Why is it called self-assembly? Because they spontaneously assemble together. All the 3 helices, they spontaneously start winding around. They spontaneously assemble together into collagen. Okay, got it? Now, after this, they have been matured in the endoplasmic reticulum. They have passed through the Golgi apparatus and they have graduated, post-graduated. Now they are mature. Now these pro-collagen, now these extension peptides will be removed. To which enzymes? Pro-collagen aminoproteinase at the amino terminal and pro-collagen carboxyproteinase at the carboxy terminal. Okay, these are the enzymes. Got it? Now we quickly move on to elastin. The function of elastin, as the name suggests, is to provide extensibility and elastic recoil to the tissues. Therefore, it will be present in tissues with maximum elasticity like lung, like large arteries. Why? Because they have to recoil after every heartbeat. Okay, after every systole, these arteries have to recoil. They have to bear the pressure of the stroke volume and then they have to recoil. Okay. Ear cartilage, elastin is present in the ear cartilage, this one. The nasal cartilage, can any of you tell me what is present at the tip of the nose? Which cartilage is this? Can you guys tell me? Elastic cartilage. This is which cartilage? Tell me in the comment section. Tip of the nose. Now, skin. Elastin is present in the skin. Obviously, skin is elastic. My skin is quite dry right now, so it's quite elastic. Anyway, now this elastin is synthesized as tropoelastin. Okay, what was collagen synthesized as? It was synthesized as pre-pro-collagen. Elastin is synthesized as tropoelastin. It is soluble. It is also present in the plasma because it is soluble. Now, when this tropoelastin undergoes a process where it is converted into an insoluble structure, that structure is called elastin and that is deposited in the extracellular matrix. Okay. It also contains proline. It also contains hydroxyproline. It contains lysine, but it does not contain hydroxylysine and glycosylated hydroxylysine. Okay. But there is an enzyme called lysyl oxidase here as well. which also causes oxidative deamination of the lysyl residues, like that was happening in collagen into aldehydes. Okay, but the cross links that were forming between two modified lysyl residues in collagen, here they are forming between a modified lysyl into aldehyde, and an unmodified lysyl. Okay, so here modified as well as unmodified lysyl are interacting together. There in collagen, both modified lysyl residues were interacting together to form interchain cross links. Okay. Now here, what are these special type of bonds called between a modified and an unmodified lysine? It is called desmosines, unique to elastin. 5-star question. Desmosines. Write a short note on desmosines. You have to write the reactions. Lysyl oxidase has the cofactor copper. You'll have to mention a word about Menkes disease as well. Okay, guys. So now quickly tell me the differences between collagen and elastin. Just the pointers. Okay, you can make a table in a tabular form. You can answer. Collagen it is synthesized as a pre-pro-collagen molecule. Okay, it is synthesized as tropoelastin, no pre-pro group. Okay, second, collagen has many subtypes 1, 2, 3, 4, 5, 6, 7, 8, 9, whereas elastin has just one subtype. Now, collagen has hydroxylysine. It does not have hydroxylysine. Collagen has glycosylated hydroxylysine. It does not have glycosylated hydroxylysine. Collagen has interchain bonds between modified lysine groups, whereas it has what? Desmosine. Got it? These are the important differences between collagen and elastin. I hope you guys have followed. If you have understood this, if you like this rapid revision, do follow me in the next class as well. That is going to happen in the next 5 or 10 minutes. Okay, I'll start. And do subscribe to this channel. Follow me on Unacademy. I'm Dr. Shivangi Shukla. You can find me under the category of NEET PG. And you'll get notifications about my different classes that I take daily. And do follow this, do subscribe to this channel, Unacademy Future Doctors on YouTube, and you'll get to see a lot of interactive, integrated videos. Very unique videos are coming up on this channel in the next year from 1st of Jan. Very unique videos, especially helpful for MBBS first and second prof students as well as NEET PG aspirants. Okay, so all of you, please guys, please subscribe to this channel and do check out all the videos. They'll be very helpful to you. You won't get such content anywhere. Okay, this is very, very helpful for you to steer you ahead of your colleagues in your classrooms. Okay, so do check out all the videos and stay tuned for very interesting stuff, very, very helpful stuff that you won't find anywhere else, and that is going to come up in the next year, this channel. All right. Bye-bye, guys. Thanks.